Hello everyone, today we will present about cell adaptation from section 2 group 1. Let's get started.
What is metaplasia? Metaplasia is the reversible transformation of one type of nature cell into another type that is better able to solve with chronic irritation or environmental stress. It is a positive and adaptive response, not a cancer. However, if the stimulus continues, it may progress to dysphasia and even cancer in the long term. Case and example: cigarette smoke can change the respiratory epithelium from columnar to squamous cell. GERD can change the esophageal lining from squamous to columnar cell, a condition called Barrett esophagus. Mechanism of metaplasia. Metaplasia occurs when stem cell or progenitor cell are reprogrammed by continuous stimulation. That cell develop into a new cell type through change in gene expression, not by direct transformation of existing cell. Key difference: metaplasia, cell type change, adaptive reversible. Hyperplasia, cell number increase, type remains the same. Dyplasia, variable cell disorganized arrangement, precancer.
Example of metaplasia in the human body. In the bronchus, urinary bladder, and salivary ducts, chronic irritation leads to a change to squamous cells. In the esophagus, acid reflux causes squamous to become columnar, known as Barrett's esophagus. Connective tissue can turn into bone with chronic trauma, and vitamin A deficiency in the respiratory tract causes squamous metaplasia. Clinical impact of metaplasia: 1. Loss of normal tissue function. The newly formed cell type often lacks the specialized function of the original tissue. 2. Risk of mutation and cancer. Persistent metaplasia under continuous stimulus and progress to dysplasia and eventually carcinoma. 3. Increased risk of infection and inflammation. The metaplastic tissue may be less resistant to pathogen or lack protective barrier. 4. Pathophysiological effect in specific organ. Eye vitamin A deficiency lead to dry eyes and risk of blindness.
Atrophies, degenerations of organs or tissues. Atrophies is a decrease in the size of cells, tissues, or organs after they have fully developed. Usually results from loss of cellular components. Mechanisms of atrophies. 1. Decreased protein synthesis. The cell does not undergo growth. 2. Increased protein degradations. Enhanced activities of the ubiquitin proteasomes pathway. 3. Autophagy. The cell degrades its own organelles in order to survive. Types of atrophies. Physiological atrophies. Uterus after parturition, smooth muscles of uterus shrinks back. Ovaries after menopause. Organ tissue shrinks. Senile atrophies. Common in aging. Pathological hyperplasias. Examples: diffuse atrophies is immobilities. Ischemic atrophies is poor blood supplies. Different from related conditions. Do not confuse with aplasia's complete absence of organ development from birth, or hypoplasia's underdevelopment of tissue organs in birth.
The last topic is what is dysplasia? Key dysplasia describes a situation where cells begin to grow and mature abnormally instead of maintaining their usual size, shape, and arrangement. The cell becomes disorganized, although it's not cancer yet. It's a warning sign that may progress to cancer if left untreated. One key point is that thus abnormal cells stay within their original tissue and have not spread to nearby areas. Key microscopic feature of dysplasia. When observed under a microscope, the plastic cells show several key abnormalities. These include nuclei that are only shaped larger than normal and more darkly stained due to HIV and E content. The nuclei also take up more space compared to the cytoplasm. In addition, we often see increased number of cell divisions, sometimes with error, and the normal alignment and structure of tissue is lost. Difference between dysplasia and hyperplasia. It's helpful to compare dysplasia with hyperplasia. Both involve a rise in cell number, but in hyperplasia, the cells look normal and are well organized. In contrast, dysplastic cells look abnormal and are disordered. Hyperplasia is usually reversible and rarely leads to cancer, while dysplasia carry a greater risk and may progress. Can dysplasia be reversed? Yes, dysplasia can be reversed, especially in its early state. If we remove the triggering factor, such as treating an infection or stopping smoking, cell can return to normal, but if left unchecked, dysplasia may progress into carcinoma in situ and eventually become invasive cancer. Common example of dysplasia. Cervical dysplasia is often caused by HPV infection. In the bronchus, smoking can lead to epithelial dysplasia. Acid reflux in the esophagus can result in Barrett esophagus with dysplasia. Lately, UV radiation can cause actinic keratosis in the skin, which is also a form of dysplasia. Diagnosis. Dysplasia is diagnosed by examining it through under a microscope, usually followed up biopsy. For cervical dysplasia, pap smear scanning is widely used. In some cases, spatial scanning or immunohistochemistry is done to investigate dysplasia from early cancer. To summarize, dysplasia is a reversible precancerous change. It shows nuclear enlargement, abnormal mitosis, and disorganized structure. If detected and managed early, it may return to normal, but if left untreated, dysplasia can become cancer. That wraps up the topic of dysplasia. Thank you for your listening and your attention.